Hey champs of Examsnet, welcome to the series of chapter-wise discussion. I am back with another session and a new chapter, that is Cell, the Unit of Life. In the previous chapter, we have studied about the structural organization in animals, and before that we studied about the structural organization in plants. So in both these chapters, we had discussed the arrangement or organization of cells to form the body of plants or animals, right? So what is the basis of these two chapters? Can we say that the basis of these two chapters is cell? Why? Because similar cells combine to form tissues, isn't it? So if a cell is not present, then can tissues be present? No, because tissues are made up of cells. So if cells are absent, then tissues will also be absent. Then if tissues are not present, then organs will also be absent, because organs are made up of tissues, and then in the absence of organ there will be no organism, because organism is made up of organs and organ system. Okay, so in the absence of organ there will be no existence of a structure or no existence of any organism. So can you see how special a cell is? So this makes it very important to have a better understanding about the cell and the components of the cell. What makes cell so important or so special? So we know that a cell is responsible for the formation of a complete structure of plant body or animal body. So we can say that this is a structural unit of life, right? Or structural unit of living organism. At the same time, it is also able to perform all the essential functions of life. So it is also a functional unit of life. So it is structural unit as well as functional unit. And we know that this is the basic requirement. Basic requirement. Everything starts from the cell. If cells will be there, then tissues will be formed, then organs will be formed, then organ system or organism will be formed. Okay, so cell is the basic or the fundamental requirement of the living organism. So if we have to define the cell, we can say it is the fundamental, structural and functional unit of all the living organism. This is the perfect definition of a cell. I hope you understood. Why structural unit? Because it forms the complete structure of the body. And why functional unit? Because it is capable of performing all the essential functions of the body, right? So from here we can conclude that cell is the basic requirement for living organism, or it is the basic requirement for life, right? At least one cell must be present if anything is to be considered a living organism. If there is no cell, then there will be no living organism, right? At least one cell must be there. For example, in unicellular organism which consist of only one cell, for example, bacteria or amoeba, right? We know that these organisms, despite having a single cell, are capable of independent existence, and they are also able to perform all the essential functions of life. So how is it so? How they are able to do these things with just a single cell? There is no organization, there is no tissues, there are no organs, just one cell. And yet they are able to perform all the essential functions, and they are able to exist independently. That means a cell is capable of doing these things, right? So how important a cell is. And obviously if the organism is multicellular, for example human body or animals, then there will be better organization of the cells, right? It will be better than that in the unicellular organisms, and the organism will be more complex or more advanced, right? But at least one cell must be present. Even if one cell is present, it will be a living object. Whereas in the living thing, and in the non-living object, there will be no requirement of cell. There is no cell present. So this is the basic difference between the living and the non-living things. The living things must have a cell, even if there is a single cell, but it should have a cell. Whereas in non-living thing, there is no cell, right? So I hope you have understood the difference between the living thing and the non-living thing. Now let's start with the discoveries of the cell. How do we come to know about the existence of cells, right? So the discoveries start from the Robert Hooke. Robert Hooke was a scientist who in the year 1665 observed the cork of the tree, right? Suppose this is the tree. He took a small part of the cork and there he observed that there are small honeycomb like structures. Okay, honeycomb like structures like this. And he named each of these compartments as cell, because in Latin language there was a word cellula, which means a small room, right? So these look like combination of small rooms, right? So there were many compartments present when you observed them through a microscope, and each of these compartments were called a cell. Okay, and the microscope that he used was also self-designed. He had designed his microscope himself. Okay, the year was 1665. But these cells were part of cork of the plant, which consists of dead cells, right? Cork of the plant are dead. Therefore, Robert Hooke observed dead cells, not the living cells. So, who observed the living cells first? It was Anton von Leeuwenhoek. The year was 1674. He observed these cells in the living organism. Okay, so which living organism he observed? He observed the unicellular organisms like protozoa or bacteria. Okay, so he observed the live cell for the first time. So who observed the dead cell first? It was Robert Hooke in the year 1665. And who observed the living cell? It was Anton von Leeuwenhoek in the year 1674. So year also you should remember, although these years are not mentioned in the NCERT. Then in the year 1831, Robert Brown observed nucleus. Okay, so these are some of the discoveries. But during these discoveries, the microscopes were not developed well, right? Because both Robert Hooke and Anton von Leeuwenhoek, they had designed their microscope themselves, both of them. And there was no such advancement in microscopes such that these small structures which are present inside the cell can be seen. They could only observe that there are cells. Okay, this was the maximum observation. But what are the things which are present inside the cells and what is going on inside the cells? These were not clear because microscopes were not well advanced. It was only in the year 1950 and after that when there was development of electron microscope. Okay, and electron microscope was a well developed microscope. Therefore, after the discovery of the electron microscope, that means after the year 1950, the details of the cells like cell membrane or ribosomes could be observed. Okay, so it was now become easy to observe the ultra structures present inside the cells or very minute organelles present inside the cell. For example, ribosome. Ribosome is very small. It could not be observed with any other cell before the electron microscope. It could not be observed by any other microscope other than the electron microscope. Okay. So this was about the discovery of the cells. Now after electron microscope, it was very easy to observe every part of the cell, including the smallest part, that is the ribosome. Now we come to the very important part of this chapter, that is the cell theory. So here there are three scientists that you have to remember. First one is Schleiden, who was a German botanist. So in the year 1838, Schleiden, whose full name was Matthias Schleiden, so in short we write M. Schleiden, he examined many plants because he was a botanist and observed that all the plants are made up of different cells which make different tissues. Okay, so he observed that the plants are made up of cells and cells are combined to form tissues. Okay. Then around the same time, just 1 year later, in the year 1839, a German botanist named as Theodor Schwann, which in short we write as T. Schwann. So he was a zoologist. So he observed the animal cells and there he observed that the cell of the animal had an outer layer or outer covering, which is now known as cell membrane. So it is today known as cell membrane, but that time this name was not given. It was just observed for the first time that there is an outer layer around the animal cell. Okay. Then he became curious. Theodor Schwann, who was zoologist, he observed that there was an outer layer around the cell of an animal. He became curious and he wanted to know whether the plant cell also has such an outer covering or outer layer. So he also tried to observe plant cell. Although he was a zoologist, but he tried to observe plant cell. Then there he was observe the cell wall, which is the outermost layer of the plant cell, right? So both cell membrane and cell wall were observed by Theodor Schwann. So both these scientists, Schleiden and Schwann, exchanged their experience, right, with each other, and then they came up together with a theory that is known as cell theory. Okay. So according to the cell theory, all the animals or plants are made up of different cells and their products. Okay, so they are made up of cells and their products. This was the cell theory which was formulated by Schleiden and Schwann together. But this cell theory did not explain how the new cells are formed. So this thing was explained by another scientist named as Rudolf Virchow in the year 1855. He used the phrase omnis cellulae cellula. In Latin language, omnis cellulae cellula. That means the cells arise from pre-existing cells. Okay. So according to him, the cells divide. So suppose this is cell number 1, this is cell number 2 that are formed after division. So he stated that these cells arise from the pre-existing cells. So cell 1 has arise from the pre-existing cell, right? And cell 2 has also the same parent cell, that was the pre-existing cell, right? So this was explained by the Virchow. Then all these scientists concluded their observations into 2 points. The first point was that all the living organisms are composed of cells and products of cells, which was given by Schleiden and Schwann. And the second point, all the cells arise from pre-existing cells, which was given by Virchow. Okay, so these two are the important conclusion about the cell, right? Now the cells of the plant body or animal body are not of the same type. They have variety of shapes and sizes, right? For example, some of the shapes and sizes are shown here. You can see RBC and WBC. Both are part of the same tissue, that is blood, right? Blood is a tissue. But RBC has different structure, different shape, and WBC has different structure, amoeboid structure, right? The structure of the RBC is round and biconcave. Similarly, columnar epithelial cells, this we have seen in anatomy of the flowering plants. These are long and narrow cells. Then we have nerve cells with cell body and dendrites. This has a completely different structure. Then a tracheid, which is part of the tissue xylem. It is an elongated cell, and then mesophyll cells of the leaves, which are round and oval. So can you see the variety in the shapes and size of the cells? So there are various type of cells. Okay, there is great diversity even in cells. Now let's have an overview of the cell. Just now we have seen different shapes of the cells. Oval shaped, round shaped, elongated shaped, etc. Similarly, the size of the cells can also vary. For example, if we take mycoplasma, what is the size of a mycoplasma? It can be about 0.3 micrometer in length. Okay. Similarly, the length of a bacteria can be about 3 micrometer. It can be about 3 to 5 micrometer length, right? Then the RBC, we have seen the shape of the RBC. It was round and biconcave in shape. Similarly, what is the size of the RBC? It is 7 micrometer in diameter. And what is the largest isolated cell? It is the egg of an ostrich. Okay, so the ostrich egg, Ostrich egg is the largest isolated cell. Other than these, you can see in the diagram given here, some type of cells are shown along with their size. So you have to keep in mind these sizes which are mentioned in the NCERT, because we cannot remember the size of all the organisms, right? So at least the ones mentioned in the NCERT must be remembered. What is the size of the virus? Here it is given 0.02 to 0.2 micrometer. PPLO, that is pleuropneumonia-like organism, it has the size about 0.1 micrometer, and the size of typical bacteria is given as 1 to 2 micrometer. But it was written that it is 3 to 5 micrometer. So it can be somewhere in between, right? We are mentioning the approximate size, not the exact size. So the exact size can be slightly more or less than the approximate size. Okay. So we have seen that there is a great variety in the shape as well as sizes of the cells. It means that different types of organisms will have different types of cells. For example, the unicellular organism will have the cell which is different from that in the multicellular organism. Prokaryotic cell will be different from the eukaryotic cell. Plant cell will be different from animal cell, right? So when we study about the cell, we broadly classify it into two category. One is prokaryotic cell and the other is eukaryotic cell. Okay, but before that, first we will be studying about the prokaryotic and eukaryotic cell separately. But before that, we will have a generalized study about the cell under this topic, an overview of the cell. Okay, so here we will have the generalized study where we will be discussing about the plant cell as well as animal cell, prokaryotic cell as well as eukaryotic cell. So first let's see the plant cell. So we have seen in the school you would have done this experiment where you were expected to observe the plant cell under the microscope, right? So what was the sample which was used to study the plant cell? The sample was onion peel cell, because it represents a typical plant cell. What did you observe? The onion peel cell, here we observed the outermost layer, that is the cell wall. Okay, so cell wall was observed as the outermost layer of the cell. Then chloroplast was observed. Central vacuole was observed, which may occupy up to 90% of the volume. So vacuole is present in animal cell as well as plant cell, but the only difference is in that in case of animal cell, the volume occupied by the vacuole is very less as compared to plant cell. In plants, the maximum volume is occupied by the vacuole. Sometimes even up to 90% of the volume can be occupied by the vacuole itself. Okay, so this makes the plant vacuole special. Then the centrioles are absent, whereas the centrioles are present in the animals. Okay, so you have to remember that chloroplast is present in the plant cell, not in the animal cell, but centrioles are present in the animal cell, not in the plant cell. Then in order to study the typical animal cell, the sample which was taken was the human cheek cell. Okay, if you remember, we used to take out or scrape out the cells from the inner cheek, okay, from the inside of our cheek using the toothpick, right, and then observe them under the microscope. So there also an outermost layer was observed, but this time the outermost layer was cell membrane, not the cell wall. Okay, so in plant cell the outermost layer is cell wall, in animal cell it is cell membrane. Then in plant cell centrioles were absent, but in animal cell centrioles are present. So these were some of the things which are specific to plant cell and animal cell. Now there are some general things which are present in both. For example, nucleus. Nucleus is must. It is the most important thing, just like the brain of our body. Just like the brain controls our body, it controls our body. Similarly, the nucleus controls the cell. Therefore, nucleus is must, whether it is a plant cell or animal cell, whether it is eukaryotic or prokaryotic. Okay. So in the nucleus contains the chromosomes, and the chromosomes contain the genetic material, that is the DNA. Okay. So nucleus will be present in both the plant cell as well as animal cell. Then the cytoplasm is again present in both the plant cell and animal cell, because all the important organelles are present in the cytoplasm. So it is the main arena of the cellular activities. All the important activities of the cells are going on inside the cytoplasm. So if cytoplasm is not present, then cell will not be able to function properly, right? So what is cytoplasm? It is a semi-fluid matrix which is present inside the cell. It occupies the volume of the cell. So suppose this is the cell. A small volume will be occupied by the nucleus. This is the nucleus. And the rest of the volume will be occupied by the cytoplasm. Okay. And inside the cytoplasm, different organelles will be present. So it is the main arena of the cellular activities. Why? Because all the important activities are performed by the organelles, and the organelles are present in the cytoplasm, right? Then it keeps the cells in the living state. If the cytoplasm is absent, then none of the important activities will take place, none of the functions will take place, so cell will not be able to function properly. Then it is universal. Universal means what? It is present in all types of cells, whether prokaryotic or eukaryotic, whether unicellular or multicellular, whether plant cell or animal cell, the cytoplasm has to be present, right? Otherwise the cell will become dead. So ribosomes. We know that there are many membrane bound organelles inside the cytoplasm, right? Membrane bound organelles found in the cytoplasm like mitochondria. Then we have endoplasmic reticulum, we have chloroplast. We will be studying about these organelles when we will discuss the eukaryotic cell. So these membrane bound organelles are found in the eukaryotic cell, not in the prokaryotic cell. Okay, so they will be absent in the prokaryotic cell. But ribosome is a non-membrane bound organelle which can be found in prokaryotic cell as well as the eukaryotic cell. Okay, so it will be found in the cytoplasm, and other than that it can also be found inside other organelles like mitochondria, chloroplast, and on the rough endoplasmic reticulum. Okay, so normally the organelles are found in the cytoplasm, but ribosome is a special type of organelle which is found in the cytoplasm. It is not only found in the cytoplasm but also in the other organelles like mitochondria or chloroplast. Also it is found on the rough endoplasmic reticulum. Okay, so there are two types of endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum. So rough endoplasmic reticulum are called rough because of the presence of the ribosomes on them. So suppose this is the endoplasmic reticulum. Okay, suppose then there can be two types of endoplasmic reticulum. In one there will be no ribosome, and in the other there will be ribosomes attached on the surface. So the presence of ribosomes will make the endoplasmic reticulum look rough, whereas if they are absent, then the endoplasmic reticulum will look smooth. Therefore the name smooth endoplasmic reticulum and rough endoplasmic reticulum is given to them. Okay, rough means the ribosome is present on them. So this is special thing about the ribosome. It is non-membrane bound organelle, that you have to remember. Mitochondria, endoplasmic reticulum, chloroplast, these are membrane bound, but ribosome is non-membrane bound. Okay, and it can be found in prokaryotes as well as eukaryotes. If they are present in prokaryotes, then they are associated with the cell membrane, and the size is about 15 nanometer by 20 nanometer. Okay. Now another important thing you have to remember is that if they are present in prokaryotes, then the ribosome is of 70S type, whereas if they are present in eukaryotes, then the ribosome is of 80S type. Okay, so 70S type and 80S type. In prokaryotes, the ribosome will be of 70S type. In eukaryotes, it will be of 80S type. But here we are talking about the ribosomes which are present in the cytoplasm. Okay, in cytoplasm they will be of 70S in prokaryotes and 80S in eukaryotes. But if they are present in other organelles like mitochondria or chloroplast, then they will always be of the 70S type. Although the organism will be eukaryote, but the ribosome will be of 70S type if they are present in other organelles. Okay. I hope you understand this. Then other important facts about the ribosome is that they are called Pallade particles because they were discovered by George Pallade in the year 1953. So the name is given after the name of the scientist. Okay, and it is also called as protein factory. Why? Because it is responsible for the translation of mRNA into protein. Right? So it helps in translation, therefore it is also known as protein factory. Polysomes, why polysome? Polysomes means many ribosomes. So a large number of ribosomes are known as polysomes. Okay. Then it is the smallest organelle, the size is 15 to 20 nanometer. Okay, 15 nanometer by 20 nanometer. This is the smallest organelle that we have to remember. And we have also discussed that it was observed only after the advent of electron microscope in the year 1950. The electron microscope was invented in the year 1950. Therefore, the ribosome was discovered after this. What was the year of ribosome? 1953. See, 1953, just after the advent of the electron microscope. Because it was impossible to observe such minute organelles using less advanced microscopes. So we have seen the generalized study of the cell, right? We have seen what are the things which are found in prokaryotic cell as well as eukaryotic cells, and what are the things which are found only in the eukaryotic cells. We have also seen some of the parts of the plant cell and some of the parts of the animal cell. Okay, for example, chloroplast is found only in the plant cell, not in the animal cell. Centrioles are present in animal cell, not in the plant cell, right? Similarly, cell wall is present in the animal cell, the plant cell, but not in the animal cell. In the animal cell, the outermost layer will be cell membrane only. Okay. Then another important thing was ribosome, which was found in both prokaryotic as well as eukaryotic. In prokaryotes, they are of 70S type, but in eukaryotes, they are of 80S type. But even in eukaryotes, if they are present inside other organelles, then they will be of 70S type. So this you have to remember. And these are smallest organelle, also known as protein factory. Now we will discuss the prokaryotic cell and eukaryotic cell separately. So now we will focus on the prokaryotic cell only first, and then after this we will discuss the eukaryotic cell. So prokaryotic cells are represented by the bacteria, blue-green algae, mycoplasma and PPLO. PPLO means pleuropneumonia-like organisms. Let's write it down here. Pleuropneumonia-like organisms. Okay, so all these are prokaryotic cells. And these cells are smaller and they multiply more rapidly than the eukaryotic cells. Okay, so the time taken for the division of cell is very less in case of prokaryotic cell as compared to that in the eukaryotic cell. Then they may vary greatly in shape and size. So you can see different shapes of the prokaryotes, especially bacteria is given. We know that bacteria can be round shaped. These are known as coccus. Bacteria known as bacillus are rod shaped. Okay. Bacteria which are known as vibrio are comma shaped. Bacteria which are spirillum are spiral shaped. Okay. So you can see different shapes of the bacteria. Size can also vary greatly. Now, although the shape and size will vary greatly, but the basic organization will be same. So the organization is fundamentally similar. Therefore, we study all the prokaryotic cells under one category. Okay. So cell wall will be present in all the prokaryotes. Okay. In the eukaryotes, it will be present in plant cell, not in the animal cell. And in prokaryotes, they will be present and they will surround the cell membrane except in the mycoplasma. So cell membrane will be present in all types of cell, okay, whether it is prokaryotes or eukaryotes, whether it is plant cell or animal cell. The only difference is that in animal cell, it will not be surrounded by the cell membrane, but in plant cell and in prokaryotic cell, the cell membrane will be surrounded by the cell wall. Then the next thing is cytoplasm. Also we have seen this is universal. It will also be found in all cells. It is a semi-fluid matrix which fills the cell or which occupies the volume of the cell, and it is the main arena of the cellular activities. Then nucleus will also be present in all types of cells, right? But the nucleus in case of prokaryotes will be without nuclear membrane. Okay, so it is not well defined nucleus. That means the nuclear membrane will be absent. Nuclear membrane is absent. Okay, so the genetic material is naked and not enveloped. So suppose this is the cell, prokaryotic cell. Inside that we have the genetic material. Normally the genetic material is enclosed within the nucleus, which is surrounded by the nuclear membrane. So here the nuclear membrane is absent. So what will happen? The genetic material will be naked. It will not be enveloped. There will be no envelope or there will be no membrane around it. Okay, so nuclear membrane will be absent. And then plasmids. What are plasmids? These are small circular DNA which are also found in the prokaryotes. These are found outside the genomic DNA. So there will be genomic DNA which is found in prokaryotes as well as eukaryotes. But in case of prokaryotes, there can be some special type of circular DNA which are found outside the genomic DNA. Okay. So two different types of DNAs can be found in prokaryotes. One is genomic DNA, that is basic, and other than that, the plasmid DNA are also found. Okay. And these plasmid DNA are extra chromosomal. That means these are found outside the chromosome, other than the chromosome, right? These are found outside the nucleus in the cytoplasm. Then they replicate independently and they confer unique phenotypic characters to the cell. For example, antibiotic resistance. Let me scroll it down. Antibiotic resistance. So this is one of the example of unique phenotypic characters which is given by the plasmids. Okay. Therefore, you will study about these in the chapter biotechnology, how important they are, right? Now cell envelope, one of the important characteristics of the prokaryotic cells. It is present in most prokaryotes, particularly bacterial cells, okay? Because mainly bacterial cells represent the prokaryotic cell, right? Now this is a chemically complex structure. It consists of tightly bound 3 layers. So it is not just one layer but a combination of 3 layers. One is glycocalyx, this is the outermost layer. Then cell wall and then plasma membrane. Okay, so the glycocalyx is the outermost layer, then the cell wall is the middle layer, and the innermost layer of the cell envelope is the cell membrane. Now you might say that the cell membrane and cell wall, these are present in eukaryotic cell as well, right? Cell wall is present in the plant cell, which is eukaryotic cell, and cell membrane is present both in the plant cell and animal cell. So how is this cell envelope different? So first difference is in the presence of the third outermost layer called glycocalyx, which is not present in the eukaryotic cell. The other and the most important difference is that in case of eukaryotes, the cell wall and plasma membrane, although they are present, but they work as different layers. Okay, the function of cell wall and cell membrane is different. They are not working as one layer. But here, all these 3 layers are working as one team. They are working as one unit. Okay, so this is the important feature of the cell envelope. So they have distinct functions, but they act together as single protective unit. The most important function of this unit is to provide protection to the cell. Okay. Let's move ahead. On the basis of difference in the cell envelope and the manner in which they respond to the staining procedure developed by Gram. So there are different types of bacteria depending on the type of cell envelope. And what is the type of cell envelope? It depends on the manner in which they respond to the Gram stain. So there is a staining procedure. staining procedure which was developed by Gram. Therefore, the stain is known as Gram stain. Okay, this is blue, violet or purple in color. Okay. Now, when the bacterial cell is going through the staining procedure, then there are two possibilities. Either the bacterial cell will take up this Gram stain or it will reject the Gram stain. If the bacterial cell take up the Gram stain, then it will appear violet or blue in color, right? Because of the color of the stain. In that case, the bacteria is known as Gram positive bacteria because it has taken up the Gram stain. The other type of bacterial cell reject this Gram stain. They do not take up this Gram stain. Then they don't appear to be blue or violet in color. In that case, they appear pink or red in color. Okay, so they go through the decolorization. So they don't take up the Gram stain. Therefore, these are known as Gram negative bacteria. Okay, so the bacteria are classified into two types: Gram positive, who take up the Gram stain, and Gram negative, which do not take up the Gram stain. Okay. Now we have seen that the outermost layer of the cell envelope is the glycocalyx. So glycocalyx is actually not having a fixed composition. The composition of the glycocalyx can vary from one bacteria to another. So it differs in thickness and composition among different bacteria. So sometimes this layer glycocalyx can be very loose and flexible. Sometimes it can be very rigid and tight. So when it is present as a loose sheath, then it is called slime layer. And when it is present as thick and tough layer or rigid layer, then it is known as capsule. Okay, so both slime layer and capsule are made up of glycocalyx only. The difference is just in the composition. Okay. Then glycocalyx was the outermost layer. The next layer inner to the glycocalyx is the cell wall. We already know about the cell wall. Cell wall is present in the plant cell also in case of eukaryotes, and in case of prokaryotes they are present in the bacterial cell. So what is the function of the cell wall? We know that the cell wall is not a flexible structure. It is a rigid structure. Okay, rigid structure. It provides protection to the cell. Therefore, it also helps in shape determination because it cannot be changed. The shape of the cell wall cannot be changed easily because it is very rigid. Therefore, it determines the shape of the cell. It also provides structural support to the cell and it prevents the cell from bursting or collapsing. Okay. So this way, the bacterial cell is prevented from bursting or collapsing in the adverse condition. Then the innermost layer is the plasma membrane. Again, we all know about the plasma membrane. It is present in prokaryotes as well as eukaryotes. It is a selectively permeable membrane. What do we mean by selectively permeable membrane? It acts as a barrier. Okay, this membrane acts as a barrier. And it selects the molecules or the materials which are to be allowed to pass through the cell and which are to be blocked. So sometimes it allows the material to pass through the cell membrane, pass through itself, and sometimes it blocks some material depending on the requirement of the materials. For example, if some materials or some molecules are beneficial for the cell, then it will identify these molecules and allow them to pass through it. Right, and if some molecules or materials are harmful for the cell, these are not required by the cell, then these molecules will be blocked by the cell membrane. That is why it is said to be selectively permeable. It selects the molecule which are to be allowed into the cell and which are to be blocked. Right, and how does the cell membrane perform this selective permeable action? So this is done by the help of the membrane proteins. Membrane proteins. These membrane proteins acts as the channel or passage for the molecules. Channels or passage. Okay. So suppose this is the cell membrane. Here we have some membrane proteins which are acting as the passage or channels for the movement. So these are the molecules which are to be passed. So they can pass through these membrane proteins. Okay. So this is how the plasma membrane selects the molecules and allows them to pass through it, or does not allow depending on the situation or requirement. Then it interacts with the outside world, and the structure is similar to that of the eukaryote. So the structure of the plasma membrane will be similar in prokaryotes as well as eukaryotes. In eukaryotes also we will be discussing about the plasma membrane. There we will have a look at detailed structure of the plasma membrane. Okay. Now modification of the cell envelope. We have seen the composition of the cell envelope, glycocalyx, cell wall and the cell membrane. We have seen the functions of the cell envelope. Now sometimes the cell envelope might get modified. And the modification of cell envelope means that there can be modification in the plasma membrane, because cell wall is also part of the cell envelope, but it will not undergo modification because cell wall is a rigid structure. Okay, it is not flexible. The flexible part is the cell membrane. So most of the modifications will be done by the cell membrane, right? So the first one is mesosome. That is nothing but the infolding of the cell membrane. So as I said that the cell membrane is a flexible structure, so it can move easily. It can extend into the cell or it can extend outside the cell, right? Both are possible with the cell membrane. So mesosomes are the extension of the cell membrane into the cell. Okay. These are specialized differentiated form of the cell membrane. And these are in the form of vesicles, tubules or lamellae. So the mesosome or the extension of the plasma membrane into the cell can be in the form of, let me draw this. This is the cell wall and this is the cell membrane, inner layer. Okay. So the extension of the cell membrane can be in the form of tubules like this. It can be in the form of vesicles like this, or it can be in the form of lamellae like this. Okay. So in any of the these manner, the cell membrane can be extended into the cell. And what is the function of the mesosome? Obviously, without importance, why will anything will be done by the plasma membrane? It has some function, it has some important role. So it has various important functions. It can help in the cell wall formation. It can help in DNA replication and distribution to the daughter cells. Then respiration and secretion as well. At the same time, we know that these are the extension into the cell. So the surface area will also increase into the cell. So the increase in surface area of the plasma membrane and enzymatic content. So mesosome is one type of extension of plasma membrane into the cell. Another type of extension of plasma membrane into the cell is called chromatophore. So when these extended plasma membrane contains some pigments, colors, then these are known as chromatophores. Okay, they contain pigments like blue-green algae. So when they contain pigments, then they give some color to the cell. Okay. Now when the plasma membrane is flexible and it can be extended inside the cell, then it can also extend outside the cell, right? Because if it is flexible, so sometimes it can extend into the cell, sometimes it can extend outside the cell also, right? So flagella is the next part, which is the extension of plasma membrane outside the cell. Okay, it will also have some important function. So let's see. It is the extension of cell membrane and from the cell wall. Okay, in the NCERT it is given that the flagella is the extension from the cell wall. So do not confuse that it is the extension of the cell wall. It is not the extension of the cell wall. Cell wall will not be extended because that is a rigid structure. Only plasma membrane will be extended from cell wall. That means it will, obviously when the cell membrane has to come out of the cell, so it will have to pass through the cell wall also, because cell wall is present outside the cell membrane as the outer layer. So it has to pass through the cell wall. That is why it is written that flagella is the extension from cell wall. It is not the extension of the cell wall. It is the extension of plasma membrane from the cell wall. Got it? And the important parts of the flagella are filament, hook and basal body. Okay. The flagella can have different parts. Let's have a look at this diagram. We have this flagella. Okay, this long portion. It is the flagella. So flagella is a long extension of the plasma membrane outside the cell. Here the longest part is the filament. Longest part of the flagella is filament. Then it has a basal body and a hook. Okay. What is the function of the flagella? It helps in motility. It provides movement to the cell. Motility or movement. So when the cell membrane is extended outside the cell, it forms flagella. It can also form some other structures, but right now we are having a look at flagella. So when it forms flagella, then it acts as the limbs of the cell. Okay, hands or legs of the cell. So it helps in motility or movement of the cell. Okay, these are long structures. Then the other type of structures that are formed by the extension of plasma membrane outside the cell are pili and fimbriae. Okay, pili are tubular structures which are made up of a protein, special protein called pilin. Okay, so pili are made up of special protein called pilin. They do not help in movement. Sometimes they might help in movement because they are hair-like structures. Okay, hair-like structures. But the most important function of pili is conjugation. They help in conjugation. What is conjugation? It is the transfer of genetic material. Transfer of genetic material, that is DNA, between the bacterial cells. Bacterial cells. So suppose this is one bacterial cell. This is another bacterial cell. So some bridge-like structure is formed between them and the genetic material is transferred to another cell. Okay, this is known as conjugation. So pili play an important role. Pili play an important role in the transfer of the genetic material. Then the third type of structure that is formed by the extension of the plasma membrane outside the cell are bristle-like fibers sprouting out of the cell called fimbriae. Okay, so fimbriae are bristle-like fibers. You can see, have a look at this figure. Pili are shown in the dark brown color and the fimbriae are shown in the pink color. These are the bristle-like fibers. Okay, flagella is also present in pink color and the fimbriae are also shown in the pink color, but flagella is long structure. Okay, and the fimbriae are small structures. Pili are also small, but these are slightly longer than the fimbriae. And what is the function of fimbriae? They help attach the bacteria to rocks in the streams and also to the host tissue. So the basic function of the fimbriae is in adhesion or attachment. So with the help of fimbriae, the bacterial cells can attach themselves to any surface. It can be the surface of hard particles like rocks, or it can be the surface of the host. Okay, it can be the host body to which the bacterial cell can attach itself with the help of the fimbriae. Okay, so let's just summarize what we have seen. We have seen that the plasma membrane can be extended. It can be extended either into the cell or outside the cell, because it is a flexible structure. Flexible. When it extends into the cell, then it can form different types of structures. For example, tubules. It can form vesicles. It can form lamellae. And if they contain pigments, then they can form chromatophores. Chromatophores. Okay, so these are the structures that are formed by the extension of the cell wall into the cell membrane into the cell. And when they extend outside the cell, then what type of structures can be formed? In that case, flagella can be formed. Then pili can be formed. Flagella. Pili can be formed. Fimbriae can be formed. Right? I hope you have understood the function of each of them. Flagella help in movement motility, pili help in conjugation, that is the transfer of genetic material, and fimbriae help in adhesion or attachment to any surface. Okay. Now let's come to the ribosomes and inclusion bodies. So ribosome we have already seen, we have already discussed the ribosome. These are non-membrane bound organelles. Non-membrane bound organelles. These are also the smallest organelles present inside the cell. We know that the difference between the prokaryotic and eukaryotic cell is that the membrane bound organelles are not found in the prokaryotic cells, whereas they are found in the eukaryotic cell. But non-membrane bound organelle like ribosome can be found in both prokaryotic as well as eukaryotic cell. Okay. When they are present in prokaryotic cell, in that case they are mainly associated with the plasma membrane. Okay, but when they are present in the eukaryotic cell, then they can be present at various locations. For example, in the cytoplasm, they can be present even inside other membrane bound organelles like mitochondria or chloroplast also. Okay, but in prokaryotes, mainly they are associated with the plasma membrane. About 15 nanometer by 20 nanometer in size. The size also we have discussed earlier. Then they are made up of two subunits. They are made up of two subunits like this. Okay, 50S and 30S, and together they are 70S prokaryotic ribosome. So we have also discussed that in case of prokaryotes, the ribosomes are of 70S types, whereas in eukaryotic organisms, the ribosome is of 80S types. Okay, 80S. Then several ribosomes may attach to single mRNA to form a chain. So suppose this is mRNA. Several ribosomes can be attached to this mRNA. Okay, so they can form a chain, and this chain of multiple ribosomes on the mRNA is known as polysome. So what is the function of polysome? We know that ribosome help in the protein synthesis. Therefore, they are also known as protein factory of the cell, right? So the function of the polysome is to increase the efficiency of protein synthesis, right? They will help in the translation of mRNA into protein. So they will increase the efficiency of protein synthesis. Got it? So we know that ribosomes are present both in prokaryotic cells as well as eukaryotic cells. But the location of ribosomes can be different, and the type of ribosome will also be different. Location in prokaryotes is the plasma membrane. It is associated with plasma membrane in prokaryotes mainly. And in eukaryotes, they are located at various locations like cytoplasm of the cell, then mitochondria, inside the chloroplast also, right? We will be discussing this when we will discuss the eukaryotic cell. And the type of ribosome will also be different in prokaryotic and eukaryotic. In case of prokaryotic cells, the ribosome will be of 70S type, whereas in case of eukaryotes, it will be 80S types. Okay, where S stands for sedimentation coefficient. Now inclusion bodies, the last thing that we have to study under the prokaryotic cells. So these are nothing but the stored reserve materials in the cytoplasm. Okay, so the reserve materials in case of prokaryotic cells are often stored in the form of inclusion bodies, and these are again not membrane bound, just like the ribosome. Both ribosome and inclusion bodies are non-membrane bound structures. Okay, that means they lie free in the cytoplasm. Then there can be various type of inclusion bodies, for example, phosphate granules, cyanophycine granules, and glycogen granules. All these are types of inclusion bodies. Then gas vacuoles gas vacuole is not inclusion body. Gas vacuole is a different thing. It is found in blue-green algae and purple and green photosynthetic bacteria. So in some bacterial cells like blue-green algae or purple and green bacteria, gas vacuoles can be found. This is the gas vacuole. So what is the function of gas vacuole? Obviously, as it is clear from the name that these are types of vacuoles in which gases will be filled, right? So what will be the function of this vacuole? This gas vacuole will provide buoyancy to the cell. Buoyancy when they are present in the water. Okay, so they will provide buoyancy in the aquatic environment. Aquatic environment. And what will be the importance of buoyancy? it will help the bacterial cell to move up and down into the water. Okay, so even if they are living in at great depth inside the water, they will still need sunlight for performing photosynthesis, right? So with the help of gas vacuoles, they can move up to the surface of the water, then they can take the optimal light required for photosynthesis. Okay, and they can also take nutrients. So this will help in maintaining their position inside the water. Okay, so this is why gas vacuoles are also important. So now we are done with the prokaryotic cells. We have seen all the important characteristics of the prokaryotic cells. Now we will have a look at the eukaryotic cells, that are more advanced form of the cells. Okay, more advanced. The prokaryotic cells were primitive type and eukaryotic cells are advanced types. One thing you would have noticed in the prokaryotic cells, that most of the components of the prokaryotic cell did not have a membrane. For example, nucleus. We know that the nucleus of prokaryotic cell does not have a nuclear membrane. Okay, so nuclear membrane is absent in the prokaryotic cells. Then membrane bound organelles. Membrane bound organelles are also absent in the prokaryotic cells, whereas non-membrane bound organelle was present. So the components of the prokaryotic cells lack membrane. So what happens when membrane is not present? What is the function of membrane? We know that membranes separate the content present inside them from the rest of the environment. Right? So inside the cell, if some components, for example organelle, contains a membrane, then this membrane will separate the content of the organelle from the cytoplasm, right? So it will create compartmentalization. Different compartments will be formed by the presence of the membranes. So since in prokaryotic cells membranes are absent, therefore compartmentalization is not present. There is a single compartment, that is the cytoplasm, and everything is lying within that compartment only, right? On the other hand, in case of eukaryotic cells, all the organelles are membrane bound except the ribosome, right? And the nucleus also contains a nuclear membrane, double nuclear membrane. So therefore, due to the presence of so many membranes inside the eukaryotic cell, there is extensive compartmentalization. So extensive compartmentalization of the cytoplasm is seen in eukaryotic cell through the presence of membrane bound organelles. Got it? Why there is extensive compartmentalization? Because of the membranes which separate the content from the cytoplasm. Then it has an organized nucleus with nuclear envelope, which was not the case in prokaryotic cell. There the nuclear materials, that is the DNA, were exposed. These were naked. Okay, they were lying naked in the cytoplasm, right? Then here the variety of complex locomotory and cytoskeletal structures will be present, that we will study. Okay, in prokaryotic cell also locomotory structure like flagella was present, but that was not a complex structure. That was a simple structure. Here more complex structures will be seen. Okay, we will study about them in detail. Then genetic material is also organized into chromosomes. I hope you know how the genetic material is organized. We will study this in class 12. We know that histone proteins are there. Histone proteins are wrapped by the negatively charged DNA molecule. Histone proteins are positively charged, and the DNA molecules which are wrapped around the histone proteins are negatively charged. Okay, so several such organizations are present, and these are present together forming a chain-like structure, that is chromosome. Okay, so this way the genetic material is organized into chromosome, and the detailed study will be done in the class 12. Now here you can see that the cells of not all the eukaryotes are same. Okay, the cells of plants are different from the cells of animals. Here we have the diagram of plant cell and animal cell. These are the diagrams which are also given in the NCERT. So have a detailed look at this diagram in your NCERT. Okay, here I am just showing this diagram. You can see that some of the components are present only in the plant cell and not in the animal cell, whereas some of the components are present only in the animal cell and not in the plant cell. For example, here you can see chloroplast is present. Chloroplast, okay, here. This chloroplast can be seen only in the plant cell and not in the animal cell. Then we have cell wall. Cell wall is also present in the plant cell, not present in the animal cell. Cell membrane will be present in both. Okay. Vacuole is present. Now vacuole will be present in both plant cell as animal cell, but in case of plant cell, the size of the vacuole will be larger than the animal cell. In plants, they can occupy up to 90% volume of the cell. Okay. Next, in the animal cell, you can see here. You can clearly see in the plant cell, the vacuole is so large, yellow color. In animal cell, there is no such large vacuole. Okay. Can you see a large size vacuole here? No, because the vacuoles are very small in size in case of animal cell. Then one of the component of animal cell is centriole, which is not present in the plant cell. Not present in plant cell. They are present only in the animal cell. So this is your task to compare the diagram of the plant cell and animal cell from your NCERT book and check which are the components present in only plant cell, which are the components only in animal cell, and see which are common and uncommon components. Okay, have a detailed study. Most of the organelles will be common in both, like endoplasmic reticulum, Golgi complex, lysosomes, ribosomes, mitochondria. All these things will be common in both. So now let's have a detailed study about each of the membranes. First one, each of the organelle. First, before the organelle, we will have a look at the cell membrane. We have studied about the cell envelope in the prokaryotes. Here, there is no such cell envelope. Here, the cell wall and cell membrane, these are present as distinct components of the cell. Okay, these are not working in a coordinated way in eukaryotes. In prokaryotes, they were working in a coordinated way. Here, they will work separately. So first, we will discuss the cell membrane and then we will discuss the cell wall. Okay. So cell membrane, it was actually the improved model of the cell membrane was given by Singer and Nicholson. Okay, Singer and Nicholson in the year 1972. So although the detailed structure of the cell membrane was already seen by after the advent of the electron microscope in the 1950, but an improved model was given by Singer and Nicholson in the year 1972. Okay, so today we study the cell membrane according to this model given by Singer and Nicholson. This is also known as fluid mosaic model. Fluid mosaic model. So everything that we will be discussing under the cell membrane will be according to the fluid mosaic model. Okay. First point is that detailed structure was seen after the advent of electron microscope, because before the advent of the electron microscope, there was the science was less advanced. Okay, microscopes were not well advanced. Therefore, these ultra structures or minute structures were not visible under those microscopes. It was only after the advent of electron microscope in the year 1950 that the detailed structures were visible. Okay, so the cell membrane was also observed after the advent of electron microscope. So using the electron microscope, we observed the cell membrane. Okay, not we, but the scientists observed the cell membrane. Then chemical studies on RBC. See, the sample that we study for cell membrane is RBC. Okay, human RBC is observed to study the cell membrane of animals or humans. So it led to the deduction of possible structure. Okay, RBC was observed. The cell membrane of the human RBC was studied to observe the cell membrane. So what did we observe? It was observed that the cell membrane consisted of lipids and proteins. So lipid was the first thing to be observed. Okay, first lipids were observed, and then after some time proteins and carbohydrates were also observed. Proteins and carbohydrates. But lipid was the first thing to be observed. Okay. The major lipids that were found were phospholipids, which were arranged in bilayer. So these lipids or phospholipids were present in bilayer. In two layers they were present. So layer 1, then layer 2. then layer 3, then layer 4. Okay, so they were present in two separate layers. Then the lipid had two parts, polar part and the non-polar part. Okay, so let me scroll down. I will explain it to you. Suppose this is the lipid. This is the polar part. Okay, this is the polar part. And this part is the non-polar part. I hope it is visible to you. Now we know that the polar molecules are hydrophilic in nature. Hydrophilic. That means they love water. They prefer aqueous environment, right? So these are present in the aqueous environment. Whereas the non-polar molecules are hydrophobic in nature. Hydrophobic. I will scroll it this side so that it is visible to you. So non-polar molecules are hydrophobic. So the non-polar part of the lipid will also be hydrophobic. It will not prefer the aqueous environment. Okay, it will prefer the non-polar environment. So if we see, this is the cell. The lipids are present in the cell membrane of the cell. So the lipids have two parts, the polar part and the non-polar part. Now the environment inside the cell is non-polar, okay, or non-aqueous environment. Inside the cell there is non-aqueous environment. Non-aqueous environment. Whereas outside the cell the environment is aqueous or polar. Okay, here we have aqueous environment. So obviously the polar part of the lipid will lie in the aqueous environment outside the cell because it will prefer the aqueous environment, whereas the non-polar part of the lipid will remain inside towards the inside of the cell membrane, and it will be protected from the aqueous environment. Got it? Two parts are there in the lipids, the polar part and the non-polar part. The polar part is hydrophilic in nature, so it will be in the aqueous environment towards the outside of the cell, and the non-polar part is hydrophobic in nature. It will not prefer aqueous environment, so it will be present towards the inside of the membrane, that means inside the cell, and it will be protected from the aqueous environment outside the cell. Okay. So it says that the polar head is towards the outside, that means towards the aqueous environment, and the hydrophobic tail. The polar part is the head and the non-polar part is the tail. So hydrophobic tail or the non-polar tail is towards the inside and it is protected from the aqueous environment. Okay. This is the fluid mosaic model of plasma membrane that was given by the Singer and Nicholson in the year 1972. Okay, so can you see the lipids, blue color and red color? You can see the blue color layer and red color layer. These are the lipids. Okay, these are present in bilayer, two different layers, blue color and red color. These are representing the two layers of the lipids. Then we have proteins. So different types of proteins are present. You can see these red color thread-like structures. These are protein. Okay. The red color structures coming out from the cell membrane, these are the proteins. And this central protein, central part that is red in color, this is also protein. So the protein are of two different types. Some proteins are coming out of the cell and some proteins are buried into the cell. Okay. So the proteins that are coming out from the surface, these are known as peripheral protein, and the proteins which are buried into the cell wall, the cell membrane, these are integral protein. Okay. So the next point is proteins and carbohydrates revealed later by biochemical investigation. So we have discussed that the lipids were first to be observed, and after the lipids, proteins and carbohydrates were also revealed by the biochemical investigation. Okay. And which cell was studied, which cell was observed? Human erythrocyte, that is RBC, was observed. Now we know that the lipids and proteins are present in the cell membrane. So the composition of the lipids and proteins might vary. Okay. That means for some cell, the protein can be present in different amount, lipids can be present in different amounts. So the amount of lipids and proteins will vary from one cell to another cell. If we talk about the human RBC that was studied to observe the cell membrane, in this case the protein is 52% and lipid is 40%. Okay, so the ratio of protein and lipid is 52 and 40. Okay, 52% protein is there and 40% lipids. So proteins are present in larger amount as compared to the lipids in human RBC. This amount can vary in different cells. Okay, now this also we have seen on the basis of ease of extraction, the membrane proteins can be of two different types, integral protein or peripheral protein. So we have seen that this is the cell membrane. Some proteins are coming out from the cell membrane like this. So these are peripheral proteins. Peripheral. These can be extracted easily. Extracted easily. Whereas some proteins are buried into the cell membrane, either partially or completely. Suppose this is the integral protein, this is partially buried because some part of the protein is also lying outside the cell membrane and other parts are lying inside the cell membrane. So this is integral protein. Some proteins can be totally buried into the cell wall like this. Okay, both are integral protein and they can be the extraction of these proteins will be difficult as compared to the extraction of the peripheral protein. Okay, so here the extraction will be difficult. Extraction is difficult. So on the basis of the ease of extraction, they can be peripheral protein which can be extracted easily and integral protein whose extraction will be difficult. Okay. Now fluidity of the plasma membrane. So the lipid that is present in the plasma membrane is quasi-fluid in nature, okay, or semi-fluid in nature. Semi-fluid in nature, which allows the lateral movement of the protein. So the proteins which are present in the cell membrane, whether peripheral protein or integral protein, these proteins can move laterally in the cell membrane and the movement will be due to the semi-fluid or quasi-fluid nature of the lipid. So this lipid will provide fluidity and flexibility to the proteins. Okay. What are the functions of the lipids? There are various functions of these lipids. They can help in cell growth, then formation of intracellular junction, secretion, endocytosis, cell division and other functions as well. Okay. So these functions were given in the NCERT. So at least these you have to keep in mind. Then the next characteristic of the cell membrane is the selective permeability, which we have already discussed. We know that the plasma membrane or the cell membrane is selectively permeable because it selectively allows the passage of some molecules in either direction. Okay, the passage or transport of molecules is not only inside the cell but also outside the cell. Okay, so it decides which molecules to allow and which molecules to not allow or which molecules to block. Okay. Now this passage will be in different way, okay, depending on the types of molecules. For example, mainly you can say that the passage of molecules will be of two types, passive and active. Passive or active. So two types of transport of molecules will take place. Passive transport takes place along the concentration gradient. Along the concentration gradient. Whereas the active transport takes place against the concentration gradient. Against the concentration gradient. We know that the environment inside the cell is non-aqueous, okay, or non-polar. So inside the cell, non-polar environment is there, and outside the cell, aqueous environment is there or polar environment is there. So suppose we have to move non-polar molecules, okay, then non-polar molecules will easily be able to transport inside the cell because the environment inside the cell is also non-polar. So like attract like, okay. So non-polar, the movement of the non-polar molecules inside the cell will be easy, okay. So it will take place through the passive transport. Okay, movement of non-polar molecules. Non-polar molecules will take place through the passive transport. It will not require energy. So no need for energy in this case. But suppose the molecules which are to be transported is polar in nature. In that case, the movement or transport of molecules will be very difficult because the environment inside the cell is non-polar and the molecule is polar. So polar sending or transporting the polar into the non-polar environment will be difficult. It will require energy. So it will take place through the active transport. Here energy will be utilized in the form of ATP. Okay, so energy is utilized for the movement of polar molecules into the cell. Polar molecules into the cell. And if the molecules are neutral, then simple diffusion will take place. So let's see what are the types of transport. First one is simple diffusion for neutral molecules. For example, water. So water will move through the osmosis. The movement of or the transport of water molecules is through osmosis. It is known as osmosis. Now passive transport and active transport. Actually osmosis also comes under the passive transport. It is a type of passive transport. Now passive transport mainly will take place when non-polar molecules have to move inside the cell, because inside the cell the environment is also non-polar. So the movement of non-polar molecules into the non-polar environment is easy. It will take place through the passive transport, whereas the movement of polar molecules into the non-polar environment will be difficult. It will require energy. So sodium potassium pump will be used for this transport and energy will be used. Okay, and this will be against the concentration gradient. Passive is along the concentration gradient and active is against the concentration gradient. So this is how selective permeability of plasma membrane operates. And this active transport will take place with the help of membrane proteins. Now let's come to cell wall. So we know that cell wall is a non-living rigid structure. Okay, it is not flexible like the plasma membrane. So because of its rigidity, it plays an important role in protection to the plants. Okay, it provides protection and structural support to the plants. So what are the functions of the cell wall? It maintains the shape of the plant, okay, or shape of the cell. Then it helps in protection from mechanical damage and infection to the cell. Here we will be talking about the cell. Okay, we are not talking about the complete plant. Then it allows cell-to-cell interaction and it also provides barrier to the undesirable macromolecules. So it does not allow the undesirable molecules or substances to move into the cell. Okay. Now composition of the cell wall. So the cell wall is composed of different things in different types of cells. For example, in case of prokaryotes, the cell wall is made up of peptidoglycans. In case of fungi, the cell wall is composed of chitin. In case of algae, it is made up of cellulose, galactans, mannans and minerals like calcium carbonate. And in case of plants, that is angiosperm plants, it is made up of cellulose, hemicellulose, pectins and proteins. So algae also come under the plant kingdom, but these are not angiosperms. Okay. So this way, the composition of cell wall varies in different cells. Then when the plant is young, then the cell wall is primary wall. Okay, in young plant the cell wall is primary wall and this is capable of growth, right? But as the cells of the wall mature, then secondary wall develops and the primary wall diminishes. So when suppose this is the primary wall, now the cells of the primary wall starts getting matured. So after they mature, during the maturation they start diminishing. Okay, the primary wall start to diminish and a secondary wall develops inside the primary wall, not outside the primary wall. Okay, so it develops inside the primary wall and later on the primary wall is removed, only secondary wall retains. Okay, so the primary wall was capable of growth, but this secondary wall will not be able to grow. Okay, it is not capable of growth, and it is formed on the inner side at maturation. This we know. Now, middle lamella is an important part of the cell wall. Suppose this is the cell wall. These are the cells of the wall. Now, these cells of the wall are held together or bound together with the help of glue-like substance, which is known as middle lamella. Middle lamella or middle lamellae. Okay, so what is the role of middle lamellae? It holds the cells of the wall together. So the substances which are present between the cells of the cell wall and helps in holding them together or binding them together is known as middle lamella. Okay, what is it made up of? It is made up of calcium pectate. It holds or glues the neighboring cells together. Okay. Now the cell wall and middle lamella may be traversed by the plasmodesmata. So sometimes there is a connection between the adjacent or neighboring cells by the plasmodesmata. Okay, suppose this is one cell, this is another cell. So a bridge-like structure is formed between these two cells to allow the movement of substances between these two cells. This bridge is known as plasmodesmata. So the cell wall and the middle lamella both will be traversed by the plasmodesmata. Okay. Now we come to the endomembrane system. So we have discussed about the plasma membrane and cell wall of the eukaryotic cell. Now we will be discussing about different organelles which are present inside the eukaryotic cell. So we know that all the organelles which are present inside the cell have distinct function and structure, right? They have different structures and different functions, but some of the organelles work in a coordinated way as a team. For example, we have seen in the cell envelope of prokaryotic cells, the cell wall, cell membrane and the glycocalyx, these were working as a team. Similarly, in case of eukaryotic cells, few organelles work together as one team. Okay, their functions are coordinated. So this system or this team is known as endomembrane system. Okay. So what are the organelles which are part of endomembrane system? First one is the endoplasmic reticulum. Endoplasmic reticulum. Then Golgi apparatus or Golgi bodies. Then the next organelle is lysosome. And the next is vacuole. Okay, vacuole is also associated with the endomembrane system. So these we will study under the endomembrane system. Let's start with the first one, that is endoplasmic reticulum. So look at the name first. Endo means inside. Endo means inside. Plasmic is related to cytoplasm. Okay, cytoplasm. So endoplasmic means inside the cytoplasm or within the cytoplasm, and reticulum means network. Network. So it was observed that some tube-like structures were forming network. Okay, so like there were tubular networks inside the cytoplasm observed by the electron microscope. So these were named as endoplasmic reticulum because these were located inside the cytoplasm and they were in the form of networks of tubules. Therefore, reticulum word was used. Okay. So they were observed under the electron microscope as or in the form of network or reticulum of small tubular structures. Okay. Now these endoplasmic reticulum are forming a vast network. So they divide the cytoplasm into two parts. One part that is present inside the endoplasmic reticulum and the other part that is located outside the endoplasmic reticulum. So they divide the intracellular space into two parts. First one is luminal part that is inside the endoplasmic reticulum, and the other part is or the other space is extraluminal space that is nothing but the cytoplasm. Okay, outside the endoplasmic reticulum. Now ribosomes are often attached to the outer surface. So sometimes the ribosomes are also, not sometimes but many times ribosomes are also seen attached to the endoplasmic reticulum. Okay. So what is the function of ribosome? Ribosome helps in the protein synthesis. Protein synthesis, right? It helps in the translation of mRNA into protein. So the endoplasmic reticulum to which ribosomes will be attached, these endoplasmic reticulum will also be involved in protein synthesis because of the presence of ribosomes, right? So this is the common sense. Now there can be different types of endoplasmic reticulum. Suppose this is one endoplasmic reticulum, this is another. We will have a look at the exact figure or a better figure. Don't take this figure seriously. I'm just using these to explain. Okay. So two types of endoplasmic reticulum are there. This is endoplasmic reticulum number 1. This is 2. In the first one, ribosomes will be attached. In the second type, ribosomes are not attached. Okay. So you can see that the presence of ribosomes on the first endoplasmic reticulum is making it look rough, right? The surface of the endoplasmic reticulum becomes rough due to the presence of ribosomes on it. Okay. Therefore, it is known as rough endoplasmic reticulum. Rough endoplasmic reticulum. So, rough endoplasmic reticulum is nothing but the endoplasmic reticulum which bear ribosomes on their surface. On the other hand, the endoplasmic reticulum which do not have ribosomes on their surface, they will appear to be smooth. They will not appear rough. Therefore, these endoplasmic reticulum are known as smooth endoplasmic reticulum. In short, it is written as SER. Okay, and the rough endoplasmic reticulum is written as RER in short. So the RER, that is rough endoplasmic reticulum, is actively involved in protein because of the presence of ribosomes on them. Okay, and they are involved in protein synthesis and secretion. Okay, then extensive and continuous with outer nuclear membrane. So the endoplasmic reticulum is in close association with the outer nuclear membrane. Okay, it is located near the nucleus. And smooth endoplasmic reticulum, which does not have ribosomes on its surface, is a major site for synthesis of lipid. So rough endoplasmic reticulum is responsible for the synthesis of protein, and the smooth endoplasmic reticulum is responsible for the synthesis of lipids. In case of animal cells, lipids like steroidal hormones are also synthesized in the smooth endoplasmic reticulum. Okay, so lipids will be synthesized in smooth endoplasmic reticulum. Here we have the figure of the endoplasmic reticulum. Can you see? This is the rough endoplasmic reticulum. You can see small ribosomes attached here. This side on the bottom we have smooth endoplasmic reticulum because no ribosomes are attached to it. These small parts are nothing but the ribosomes. And as you can see that this endoplasmic reticulum is located near the nucleus. It is attached with it is continuous with the outer nuclear membrane. Okay, this central part is the nucleus. Okay, so this is the figure of endoplasmic reticulum. Now the next component of the endomembrane system is Golgi apparatus. As we have discussed, their functions will be coordinated. They will work as a team. So what is the function of endoplasmic reticulum? It helps in synthesizing proteins and lipids, right? The rough endoplasmic reticulum, protein, and smooth endoplasmic reticulum, lipids. Now these materials which are manufactured by the endoplasmic reticulum will be now packaged by the Golgi apparatus. So it helps in the package of the materials which will be delivered to their targets, whether intracellular targets or extracellular targets. Okay, it is similar to something during shopping. Suppose you do online shopping. You do online shopping. So what are the stages of the materials to that come to you? First, these materials are manufactured, right? These materials are manufactured at their centers, manufacturing centers. Then these materials which have been manufactured are sent to the other centers where they are packaged. So after manufacturing, packaging takes place, and after packaging the materials are delivered to you. Okay, so after packaging, delivery takes place. So these are the three main steps: manufacturing, then packaging, and then the delivery. Okay, so the manufacturing in this case, in the case of eukaryotic cells, takes place by the endoplasmic reticulum. Okay, manufacturing takes place in the endoplasmic reticulum. Here we are talking about the manufacturing of lipids and proteins. Then these manufactured materials will be packaged by the next organelle, that is the Golgi apparatus. So the main function is packaging. This organelle was observed by Camillo Golgi in the year 1898 as densely stained reticular structures near the nucleus. Again, these are reticular structures. That means network-like structures, and these are also present near the nucleus because endoplasmic reticulum is located near the nucleus and Golgi apparatus is located near the endoplasmic reticulum, right? Because they have to work in a coordinated way. So both are present near the nucleus. What is the structure of the Golgi apparatus? They contain many flat disc-shaped sacs known as cisternae. Okay, so like this, disc-shaped sacs are there. These are known as cisternae. This is one cisternae, this is another cisternae. As you can see that the cisternae has two faces. One is convex face. This you can see this is outer convex face and the inner concave face. You understand convex and concave? Suppose this is the curved part. So the outer part is convex and the inner part is concave. Okay, this you will study in physics. You would have studied in junior classes as well. So cisternae also have two faces, convex, outer convex and the inner concave. Okay. The name will be different. The outer convex surface is known as cis and the inner concave surface is known as trans. Okay. So the cisternae are 0.5 micrometer to 1 micrometer in diameter. These are stacked parallel to each other. You can see one cisternae, then another cisternae, then another cisternae. So these cisternae are stacked parallel to each other, and the number of cisternae is not fixed. They can vary. Okay, these are arranged concentrically near the nucleus. Concentrically means their center will be common. So see, this is the center. You can see for this cisternae also this is the center. For the next cisternae this is the center. So the center is common. Okay. Suppose this is the circle. So they have the same center. This is known as concentric arrangement. Okay. They have two surfaces we have already discussed. A convex surface, the outer convex surface which is known as cis, and the inner concave surface which is known as trans, and this is the maturing surface. Okay. Or the maturing face of the cisternae. So two faces are there, cis face and trans face. Cis face and trans face. Now when the manufactured materials come from the endoplasmic reticulum, they enter to the cis phase. Okay, that means the convex phase. This is the convex phase and here we have endoplasmic reticulum. They enter through the convex phase, and after packaging takes place inside the Golgi apparatus. They are released from the trans phase or the concave phase. Okay, so they will enter through the convex phase known as cis phase and they will be released after packaging through the trans phase or the concave phase or the maturing phase. Okay, here we have the diagram. You can see these cisternae, right? These are stacked parallel to each other. Now what are the functions? We have already discussed. The main function is in packaging materials. Okay, they help in packaging the materials. So the materials which come from the endoplasmic reticulum enter to the cis phase, that is the convex phase of the cisternae, and then after packaging they are released from the trans phase. Okay, so this sequence you have to remember. Then they also help in the modification of the protein which is synthesized by the ribosomes on the endoplasmic reticulum, and also formation of glycoproteins and glycolipids. So they also play an important role in the formation of glycoproteins and glycolipids. Next organelle, which is again part of the endomembrane system, is the lysosome. It is also membrane bound vesicular structure. So all the organelles that we are studying are membrane bound. Okay, so lysosome is also membrane bound. It is formed by the process of packaging in the Golgi apparatus. Okay, How is it formed? It is formed by the process of packaging in the Golgi apparatus. So this is how it is related to the Golgi apparatus. Now this organelle is rich in hydrolytic enzyme. This is the most important characteristic of lysosomes. Okay, it consists of hydrolytic enzymes. What is the function of hydrolytic enzyme? It helps in digestion. Hydrolases like lipases. For example, lipases. So what is the function of lipase? It helps in digesting the lipids, digesting or breaking down the lipids. What is the function of proteases? It helps in digesting the proteins. Similarly, there will be different hydrolases to digest different components of the food like lipids, proteins, carbohydrates, etc. So what is the main function of lysosomes? It helps in digestion. Okay. And this enzyme is optically active at acidic pH. Right. So the function of the lysosome is the digestion. Sometimes what happens, it digests the complete cell because it digests all the components present inside the cell. So the complete cell is digested, which leads to the collapse of the cell or death of the cell. Therefore, the lysosome is also known as suicide bags or suicide sacs. Okay, so this is important about the lysosomes. Next is vacuole. Vacuole is also membrane-bound space in the cytoplasm. It consists of single membrane, which is known as tonoplast. Okay, it contains water, sap, and waste material. This tonoplast, that is the membrane of the vacuole. The vacuole of the plant can occupy 90% space in the plants, whereas in case of animals, the little space will be occupied by the vacuole. So the size of the vacuole will be smaller in case of animal cell, and it will be very large in case of plant cells. Okay. Transport of ions and other materials against the concentration gradient. Okay, so the vacuoles allow this single membrane tonoplast allows the movement of the ions and other materials against the concentration gradient. That means the concentration of the materials which are being transported, for example ions, will be slightly higher in the vacuole as compared to that in cytoplasm. So the concentration will be lower in the cytoplasm. Got it? So the movement of materials is taking place against the concentration gradient. Now, some special type of vacuoles like contractile vacuole are present in amoeba, which help in osmoregulation and excretion. So the main function of contractile vacuole is osmoregulation and excretion. Similarly, there is food vacuole, which is present in protists and other cells, and it is formed by engulfing the food particles. Okay, so these were parts of the endomembrane system. Now there are other organelles also like mitochondria, chloroplast, but these are not part of the endomembrane system. Okay, these will work in a separate manner. Okay, their actions will not be coordinated with other organelles. So let's start with the mitochondria. So mitochondria, this is another important organelle of the eukaryotic cell. Okay. Now, mitochondria is a plural form. The singular is mitochondrion. Mitochondrion. Okay. The number of the mitochondria will be variable in different cells. Its number depends on the physiological activity of the cell. So, the cells which are physiologically more active will have more number of mitochondria in them. Okay. What is the shape of the mitochondria? It is typically sausage or cylindrical shaped, although the shape or size of the mitochondria will also vary considerably, but typically it will be sausage shaped or the cylindrical shape. The size is 0.2 to 1 micrometer in diameter. So here we are talking about the diameter. 0.2 to 1 micrometer, and average will be 0.5 micrometer. And the length will be about 1 to 4 micrometer. Okay, so this you have to remember. Now this is double membrane bound structure or double membrane bound organelle. So it will have an outer membrane and an inner membrane. Okay, so the outer membrane will form a continuous limiting boundary. So suppose this is the outer membrane, so it will form a continuous limiting boundary like this, and the inner membrane will make a number of infoldings known as cristae. So this is the outer membrane, right? And now we will draw the inner membrane. So this is the inner membrane like this, right? So can you see? The inner membrane is making a number of infoldings, and these infoldings are known as cristae. Okay, what is the function of cristae? They increase the surface area. We know that the infoldings of membrane will increase the surface area, right? And the inner membrane will be filled, the inner part will be filled with a dense homogeneous substance known as matrix. Okay, so what we saw that the mitochondrion consists of two membranes and an outer membrane which forms the limiting boundary of the organelle, and the inner membrane which forms a number of infoldings known as cristae. Okay. Now these two membranes are dividing the lumen of the mitochondria into two compartments, and an outer compartment and an inner compartment. So this is the outer compartment which is present between the outer membrane and inner membrane, and this is the inner compartment that is present inside the space that is limited by the inner membrane. Okay. So outer compartment is the space between the outer membrane and the inner membrane, and inner compartment is the space that is limited by the inner membrane. Got it? And the inner compartment is filled with dense homogeneous substance known as matrix. So the substance that is present is known as matrix, and the infoldings made by the inner membrane are known as cristae. Okay. Now this mitochondria is site for aerobic respiration. Aerobic respiration takes place in the mitochondria. It results in the production of cellular energy in the form of ATP. Therefore, the mitochondria are also known as powerhouses of the cell. Okay, because the production of energy takes place in the mitochondria in the form of ATP. Okay. Now, we saw that the space that is limited by the inner membrane is filled with matrix, right? So the matrix consists of many things like single circular DNA molecule. Okay, this is the outer membrane, this is the inner membrane, right? Like this. So inside the inner membrane we have matrix, and the matrix consists of single circular DNA molecule. Then it also consists of few RNA molecules. It can consist of ribosomes, and this ribosome will be of 70S type. Again I'm saying that if the ribosomes are present inside the organelles, it will be of 70S type, and if it is present in the cytoplasm, it will be of 80S type. Okay, and other components required for protein synthesis. Okay, so these things will be present inside the matrix of the mitochondria. How does the mitochondria divide? It divides by fission. That means simple breaking into two parts. Okay. Here we have the figure of mitochondria. Diagram is there. You can see an outer membrane is there, an inner membrane is there. Then the green color space is limited by the inner membrane, is known as matrix. Okay, and then crista, that means the infolding made by the inner membrane. Okay, so this is the mitochondria. Now we come to the next organelle, that is plastids. Now one of the important differences between mitochondria and the plastids is that the mitochondria are very small in size. Okay, these are very small. So the mitochondria are very small, so they have to be specifically stained. Okay, they have to be stained specifically, then only they will be visible under the microscope. Otherwise, if they are not stained, it will be very difficult to observe the mitochondria even under the microscope. Okay, so the staining is must to observe the mitochondria. On the other hand, the plastids are large in size. They can be easily observed. They can be easily observed under the microscope even without staining. Okay, so this is the difference between the mitochondria and the plastids. The size is different. Mitochondria is very small, the plastids are larger in size. These are present in plant cells and euglenoids. Now the plastids contain different types of pigments which impart different colors to the plant. Okay, the pigments impart different colors to the plants. Now depending on the pigments, depending on which pigment is present in the plastids can be of different types. For example, chloroplast. So chloroplast is a type of plastid which contains the chlorophyll and carotenoid pigments. Okay, so which pigments are present in the chloroplast? Chlorophyll and the carotenoid pigments, which are essential for trapping light energy. Okay, so they will help in trapping the light energy. Majority of the chloroplast are found in the mesophyll cells of the leaves. Mesophyll cells of the leaves. Okay. Now the number of chloroplast can also vary. For example, in Chlamydomonas, the number of chloroplast is 1 per cell. So in one cell of the Chlamydomonas, only one chloroplast is present. Whereas in case of mesophyll cells, there are large number of chloroplast. It can be about 20 to 40 chloroplast per cell. Okay, so the number of chloroplast varies from cell to cell. Okay, now the next type of plastid is chromoplast. So we are classifying the plastids on the basis of the pigments present in them, right? So in case of chloroplast, the pigments were chlorophyll and carotenoid pigments. In case of chromoplast, which pigments are present? So here carotene, xanthophylls, etc. are present, which are fat soluble. Okay. Then the carotenoids, xanthophylls, these pigments impart yellow, orange or red color to the plant. Okay, so if chromoplast is present as the plastid, then the plant will appear to be yellow, orange or red in color. Then the next type of plastid is leucoplast. Leucoplasts are colorless plastids. They do not possess any color. Okay, because they do not have the pigments that impart color to the plants. Instead, they have stored nutrients. So the main function of leucoplast is to store the nutrients. Now again, leucoplast can be of different types depending on which nutrient is stored. For example, carbohydrate is stored or protein is stored. Okay, so if carbohydrate is stored in the leucoplast, then this leucoplast is known as amyloplast. Okay, if the carbohydrate or starch is stored, then it is known as amyloplast. If the stored nutrient is oil and fats, okay, if oils and fats are stored in the leucoplast, then this type of leucoplast is known as elaioplast. And if the stored nutrient is protein, then it is known as alluroplast. Okay, so the leucoplasts are also of different types depending on which nutrients are stored. If carbohydrates are stored, then it is known as amyloplast. If oils and fats are stored, then it is elaioplast. And if it is protein, if protein is stored, then it is alluroplast. Okay. Now here we have the shape and size of chloroplast. Okay, this is shape and size of chloroplast. Because chloroplast is the most important plastid, because these are present in maximum amount in the plants. Okay, all the plants look green in color. They appear to be green because of the presence of chloroplast. So this is the most important plastid. So what is the shape and size of the chloroplast? It can be lens shaped, oval, spherical, discoid or ribbon-like. Okay, the length of the chloroplast can be 5 to 10 micrometer and width can be 2 to 4 micrometer. Okay, so this you should remember. Again, the plastids are also double membrane bound structure, just like the mitochondria. Mitochondria also consisted of double membrane. Here also double membranes are present. So an outer membrane will be there and inner membrane will be there, out of which the inner membrane is less permeable and it surrounds the stoma. Stoma, what is stoma? It is the space. It is the space that is limited by the inner membrane. Inner membrane. And what is the name of the space that is limited by the inner membrane of the mitochondria? That consisted of matrix. Okay, matrix were present. Here we have stroma. That space is known as stroma. Now, what are the composition of the stroma? What are the things that are located in the stroma? So, thylakoids. What are thylakoids? These are organized flattened membranous sacs like this. Like this, okay? Organized flattened membranous sacs. So, suppose this is a coin. We put coin one on the other, right? We make the piles of coins, okay? Similarly, thylakoids are also arranged inside the plastids. Okay, so these are organized flattened membranous sacs which contain chlorophyll pigments. Okay, and these are arranged in stacks, and the stacks are known as grana. So these are arranged just like the piles of coins. Now grana, what is grana? Suppose this is one thylakoid. This is one thylakoid. Then we have another thylakoid. Then another thylakoid. So different thylakoids are arranged on the top of other. Now all these thylakoids together are known as grana. Okay. So this pile that is made by arrangement of thylakoids one on the other, this pile is known as grana. Now what is stroma? Lamellae. These are flat membranous tubules which connect the thylakoids of different grana. So I have explained that the thylakoids are stacked one on the other, and this arrangement together is known as grana. So many such grana will be present inside the stroma. Okay, suppose this is the stroma. This is the stroma. That is the space occupied by the limited by the inner membrane. Here we have one thylakoid, on the top of it another thylakoid, on the top of it another thylakoid. Okay, so these thylakoids together are making grana. So this pile of thylakoid or stacks of thylakoid is known as grana. Similarly, we have another grana here, one thylakoid on the other, again another grana, thylakoid upon thylakoid. Now the thylakoids of the different grana can be joined together with the help of stroma lamellae. So stroma lamellae are the membranous tubules which connect the thylakoid of different grana together. Okay. Then contents of stroma, other things that are present inside the stroma are enzymes which are required for the synthesis of carbohydrates and proteins. Then small double standard circular DNA molecule will be present, and some ribosomes can also be present. Again, we know 70S ribosome will be present, not 80S. Okay. In case of mitochondria also, the two membranes, the outer membrane and inner membrane, consisted of different enzymes which were associated with the mitochondrial functions. Okay. Here the enzymes are located or present in the stroma. Now we have the diagram of chloroplast. Already I have explained, but still let's see. Let me zoom in. Can you see? Small ribosomes are also present in the stroma, which are 70S ribosome. Then these are the thylakoids. Again, this is another thylakoid. This is another thylakoid. Thylakoid above the thylakoid, and these are together known as the grana. Okay. These are together known as grana. And the thylakoids of different grana are connected with the help of stroma lamellae. Okay. So I hope the figure is clear to you. Now we move to the next organelle, that is the ribosome. The mitochondria and plastid were membrane bound organelles, okay, double membrane, whereas ribosome is non-membrane. We have studied ribosome under the prokaryotic cell as well, and here also we will study. The only difference is that the ribosome in case of eukaryotes will be of ATS type if they are present in the cytoplasm. And if they are present in the organelles like mitochondria or plastids, then they will be 70S. Okay. In the cytoplasm, they will be ATS. These are granular structures observed under the electron microscope. These were discovered by George Palade in the year 1953. Therefore, these are also known as Palade particles. Okay. After the name of the scientist. These are non-membrane bound. No membranes are present. Therefore, these are also found in prokaryotic cells. These are made up of RNA and protein. Okay, this you have to remember, these are made up of RNA and protein. In case of eukaryotic cell, these are of 80S type. In prokaryotic cell, these are of 70S types. S stands for Swedberg's unit, okay, or sedimentation coefficient. Can you see here? We have studied that the ribosome consists of two subunits. So we have two subunits here. One is small subunit and the other is large subunit. Okay, so this is the ribosome. So we don't have to go into detail of ribosome because we have discussed about it again and again, okay, in different parts of the chapter. Now cytoskeleton. Cyto means cell and skeleton. What is skeleton? It is the supportive structure, okay, just like the skeleton of your body which provides support to the body, and at the same time the skeleton also helps in the movement of the body, right? So the cytoskeleton of a cell also plays the similar function, that means the support and the motility. So what is cytoskeleton? It is an elaborate network of filamentous proteinaceous structure in the cells. These are present in the form of microtubules, microfilaments and intermediate filaments. Okay, so cytoskeleton consists of microtubules, microfilaments and intermediate filaments. What is the function of cytoskeleton? It provides mechanical support, motility and maintenance of shape, okay, of the cell. So mainly there are different types of cytoskeleton like cilia, flagella. These are the type of cytoskeleton only, okay. Other than that, we have centrioles and centrosome. Okay, so these are examples of cytoskeleton. Let's first discuss the cilia and flagella. So what are cilia and flagella? These are the hair-like outgrowths of the cell membrane. We have already discussed that cilia and flagella are the modification of the plasma membrane only. Okay, these are the extension of the plasma membrane outside the cell. Okay, whereas there were chromatophores and tubules, vesicles, etc. that were the modification of plasma membrane or extension of plasma membrane into the cell. Okay, these were known as mesosomes. So mesosomes and chromatophores were the extension of plasma membrane into the cell, whereas cilia and flagella, these are the extension of plasma membrane outside the cell. Okay. So these are hair-like outgrowths. The difference is that cilia is smaller in size, whereas flagella are longer in size. So the number of flagella is also few and the number of cilia is large. Okay. So many cilia are present. These are smaller in number. There are many cilia. Suppose this is a cell, then many small hair-like structures will be present. These are known as cilia. Okay. They help in movement of the cell. So they can either move the cell or they can move the surrounding fluid just like the oars. Okay, so they can work like oars and they can move the cell or either the cell or the surrounding fluid. Okay, so this way they are helping in the movement of the cell. On the other hand, flagella is longer in size and there are few flagella. For example, this is a cell. So one or two flagella can be present. These are longer in size. Okay. They also help in the cell movement. Now the structure of cilia and flagella are similar. Okay. The core is known as axoneme. See, let's have a look at the transverse section of cilia or flagella. It is like this. Okay. The shape is cylindrical. It looks cylindrical like this. Okay. So if we take the transverse section, it will look circular like this. So we are having a look at the transverse section of the cilia or flagella. The core is called axoneme. Now what are the things that are present in the axoneme? So it consists of 9 plus 2 array. Okay, what is 9 plus 2 array? So there are many microtubules present inside the axoneme, okay, inside the core. There are microtubules, number of microtubules which are running parallel to the long axis. So this is the cylindrical part, the structure of the cilia or flagella, cylindrical. So here we will have the microtubules which will be running parallel to this axis, long axis, like this. One microtubule here. Got it? Now if we have a look at the transverse section, we can see that we have doublets of microtubules. Okay, so the microtubules are present in the form of doublets or couples. Okay, like this. Two microtubules together forming a doublet. Again, two microtubules together forming a doublet. Two microtubules together forming a doublet. Again. So total 9 such doublets will be present. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so 9 such doublets of microtubules will be present. Other than these, there will be 2 microtubules at the center. Okay, so 9 doublets at the periphery and 2 microtubules separately at the center. Okay, these two will not be present in the form of doublet. The central microtubules are not present in the form of doublets. They are present separately, but they will be connected by a bridge. Okay, and these two will be enclosed by a sheath called central sheath, right? So this arrangement is known as 9 plus 2 arrangement or 9 plus 2 array. 9 means 9 doublets are there at the periphery and 2 means 2 microtubules are present at the center. Okay. Now the doublets are connected among themselves and these doublets are also connected to the central sheath. So suppose this is one doublet, this is another doublet. So these two doublets are connected by inter-doublet bridge. Okay, inter-doublet bridge. All the doublets will be connected by the inter-doublet bridge. And then these doublets will also be connected with the central sheath with the help of radial spoke. Okay. So inter-doublet bridge connects the adjacent or neighboring doublets. Okay, connect the adjacent doublets, whereas the radial spokes will connect the doublet to the central sheath. Connect the doublet to the central sheath. Okay, so have a look at this diagram. You can see 9 doublets are there. 1 doublet, 2, 3, 4, 5, 6, 7, 8 and 9. Okay, these are connected with each other with the help of inter-doublet bridge. Okay, then at the center we have 2 microtubules, central microtubules, which are connected again by with the help of a bridge, and these are enclosed within the central sheath. Now the peripheral doublets are connected with the central sheath with the help of radial spoke. Okay, this is the radial spoke. So total 9 doublets are there. So how many radial spokes will be there? The 9 radial spokes will be there. Okay, connecting each of the doublets to the central sheath. So I hope you have understood this. All are enclosed within the plasma membrane. Okay, these are surrounded by the plasma membrane. So this is the structure of the cilia and flagella. Now let's come to the centriole and centrosome. You would have heard of centriole and centrosome, that these are found only in the animal cell. These are not found in the plant cell. Okay. Now here I think I don't need to mention the diagram of centriole. It is similar to the cilia and flagella. The only difference is that in case of centriole there are triplets, not doublets. Okay, triplets. In case of cilia or flagella, there were doublets. Okay, microtubules were present in the form of doublets. Here, the tubulin will be present in the form of triplets. That means 3 tubules together. Okay, 3 tubules together. Right? Like this. And another difference will be that in case of cilia and flagella, there were microtubules also present at the center. But here at the center, there will be no microtubules. Okay, so no microtubules at the center. No microtubules at center. So what will be present at the center? It will be a proteinaceous material. Proteinaceous material known as hub. So proteinaceous hub will be present at the center of centrioles. Okay, and here also structure will be cylindrical like this, just like the cilia or flagella, right? Then what is centrosome? Centrosome consists of two centrioles which are arranged together at an angle of 90 degree. Okay, so when two centrioles are arranged at an angle of 90 degree like this, then they are surrounded by amorphous pericentriolar material, then this forms the centrosome. Okay, so it consists of two centrioles which are arranged like a cartwheel. Okay, so they have an organization just like a cartwheel, and they are surrounded by amorphous pericentriolar material. Okay, pericentriolar material. Here we have the structure of the centrosome. You can see this is one centriole. This is another centriole. These are cylindrical in structure, and can you see the triplets here? Triplets are present. Okay. These are arranged at an angle of 90 degree. So this forms the centrosome, and it is found only in the animal cell. Now we will move to the next topic. Now we have come to the last part of the chapter and the most important part, that is the nucleus. Okay, so nucleus is very important because it's just like the brain. Okay, as the brain controls the function of our body, similarly the nucleus controls the function or the activity of the cell, right? It was first discovered by Robert Brown in the year 1831. Later on, Fleming stained the nucleus and he observed an elaborate nucleoprotein fiber known as chromatin. Okay, so actually chromatin name was not given earlier. Fleming gave this name. Okay, when Fleming observed the nucleoprotein fiber after staining the nucleus, then he named this fiber chromatin. So the name was given by the Fleming. Okay. Then it is a double membrane bound structure. So nuclear membrane consists of nuclear envelope consists of two membranes, an outer membrane and an inner membrane. So suppose this is an outer membrane, this is the inner membrane. So there is some space between two membranes. This space is known as perinuclear space, which is about 10 to 50 nanometer. Okay. Now this nuclear envelope acts as the barrier between the contents of the nucleus and the cytoplasm. So what is the function of a membrane, nuclear membrane or any envelope? The function is to protect the inner environment from the outer environment. Okay. So similarly, this nuclear membrane also protects the nuclear content from the outer environment, that is cytoplasm. It keeps the nuclear content separate from the cytoplasm. Okay. Now have a look at this diagram. Here you can see endoplasmic reticulum. We have discussed the organelle endoplasmic reticulum. There also we had seen that the endoplasmic reticulum remains in close association with the nucleus, right? So it is continuous with the outer nuclear membrane. So you can see that the outer nuclear membrane is in continuation with the endoplasmic reticulum. It continues with the endoplasmic reticulum. And the inner nuclear membrane is in contact with the inner components of the nucleus. Okay. Now sometimes the two membranes of the nucleus, that is the outer membrane and the inner membrane, can fuse together. Okay. Suppose this is the outer nuclear membrane, this is the inner nuclear membrane. Sometimes they can fuse together like this. So when they fuse together, at that location a small pore is created. So these are known as nuclear pores. So you can see nuclear pores are also shown in this diagram, and these are also very useful because these pores are used for the transportation of RNA and protein into and outside the nucleus. Okay, so the RNA or protein can be transferred in both the direction, okay, through these pores. Then the nuclear matrix is present, matrix inside the nucleus. This is known as nucleoplasm. What is nucleoplasm? This is the nuclear matrix. And then in the nuclear matrix, small spherical bodies can be seen, which are known as nucleolus. Nucleolus is the singular form and the plural is nucleoli. Nucleoli. Okay. These are present in the nucleoplasm, and these are active site for ribosomal RNA synthesis. Ribosomal RNA synthesis. Okay, so when you have a look at an interphase nucleus, interphase nucleus. Why interphase nucleus? Because this is the phase when the cell is not dividing. So no cell division is taking place during the interphase, right? So if you look or observe the nucleus of an interphase nucleus, then you will see the components like chromatin. Chromatin will be visible. Then the nuclear matrix, that is the nucleoplasm, can be seen. And inside the nucleoplasm, the small spherical bodies known as nucleoli, these are also observed during the interphase. Okay. And when the cell starts dividing, then in place of chromatin, you will see chromosome. Okay, structured chromosome will be visible during the cell division. Okay, so when you observe the nucleus of a dividing cell, in that case, what will be observed? Chromosome will be observed in place of the nucleus or in place of the chromatin. So the chromosome consists of two sister chromatids which are attached together at the centromere. Okay, so you can see two sister chromatids are there in this figure. And the chromosome has a primary constriction called centromere. Okay, what is centromere? Centromere, this is the primary constriction of the chromosome. Primary constriction. And on the sides of the centromere, two disc-shaped structures are found which are known as kinetochores. So kinetochores are shown in this diagram, right? So this is the figure of chromosome with the kinetochore. And you have to remember that the chromosome will be visible only in a dividing nucleus, okay, or dividing cell. When the cell is not dividing, then chromosome will not be visible, only chromatin will be visible, okay. Now, depending on the position of the centromere, the chromosome can be of different types. For example, you can see in the first one here, 4 chromosomes are shown. This is 1, this is 2, this is 3, and this is 4. So in the first chromosome, the centromere is located at the center. So this is the upper arm of the chromosome, and this is the lower arm. So in the first case where the centromere is located at the center, then the length of the upper arm and the lower arm are equal because the centromere is dividing the chromosome into 2 equal parts. Okay, so this is known as metacentric chromosome. Okay. In the next case, in the next one, you can see that the centromere is located slightly above the center. Okay, slightly away from the center. So it is making one short upper arm. So the upper arm is shorter and the lower arm is longer. Right? This is known as submetacentric chromosome. So gradually the position of centromere is moving, okay, is changing. The centromere is moving upwards. In the third one, again the centromere further moves up and it is located at such a position where the upper arm is very small and the lower arm is very long. Okay, this is known as acrocentric chromosome. And in the last one, the centromere is terminal. Okay, terminal centromere. So this is known as telocentric chromosome. So how many types of chromosomes are there on the basis of the position of the centromere? Metacentric, then submetacentric, then acrocentric, and then telocentric. Okay. So this was all about the nucleus and the chromosome. We will be discussing more about chromosomes and the division of the nucleus or division of the cell in the chapter Cell Cycle and Cell Division. Okay. Now other than this, you can remember one thing, that sometimes the chromosomes can have secondary constriction as well. Okay. We have seen that the centromere is the primary constriction. Sometimes secondary constrictions can also be present. Secondary constrictions. And when these constrictions are present, these are present at constant distance. Okay. At constant interval. So they look like fragments. They give the appearance of a fragment. Okay, so you can see at the top of the first chromosome, this is shown secondary constriction. These are known as satellite. Okay, secondary constriction are known as satellite. Now, sometimes in both plant cell as well as animal cell, small membrane bound vesicles can also be found. Membrane bound vesicles. These vesicles are not present in the nucleus. We have completed the topic nucleus. Other than the nucleus, I am saying that sometimes in both plant cell and animal cell, small membrane bound vesicles can be found in the cytoplasm. Okay, these vesicles contain enzymes, and these are known as microbodies. Okay, so microbodies can also be present in the plant cell and animal cell. It is not compulsory that it will be present. They can be present or they cannot be present. Okay. So we have completed this chapter. I hope you have understood this chapter well. If you have any doubt, please feel free to mention your doubts in the comment section. I will be replying to all your comments. Okay. So we will be back with another session. Till then, keep learning, keep scoring high. Thank you so much.